Hi, I would like to welcome everyone to this week's edition of Extracellular Vesicle Club. EVClub was established by Ken, and it has now grown into an event of the International Society for Extracellular Vesicles. My name is Metka, Lenassi, and I'll be your host for today's event, where we will discuss about supermeres and exomeres and their relation to extracellular RNA and EVs. You might have read the recent communication instigated by Ken Witwer and Robert Coffey in the Journal of Extracellular Biology. And to continue those interesting discussions, it is my great pleasure to welcome the authors of the mentioned exchange, Robert Coffey, Qin Zhang, Dennis Jeppesen, Juan Pablo Tosar, and Alfonso Cayota and Ken Witwer. And to get started, Robert will give a bit of the background, after which Juan Pablo and Robert will each present their view.
Okay, I want to thank Ken for organizing this session. And we're going to be doing a bit of tag team here. So the focus is this article that we published in December in Nature Cell Biology. And then there are dueling commentaries, if you will. And I have to say that that the exchanges we've had related to these two commentaries with Ken and Juan Pablo have been, for me, a model of scientific discourse. The exchanges have been highly collegial and issue-oriented. So the way we were hoping to organize our part of the presentation today was to have both Dennis to start off, and he'll give a brief background on EVs and nanoparticles, and then Chen will delve deeper into the specifics of our paper. And I want to mention that this work has been supported by the Extracellular RNA Communication Consortium that Ken and Pablo are also part of. And I'm PI along with Al Sharest, and this work has been carried out within the Epithelial Biology Center. So now I'm going to turn it over to Dennis.
Okay, thank you for that introduction, Bob. And I think that this audience today is well aware that in the recent years, we have had a real explosion in the number of different types of extracellular vesicles and nanoparticles that has been introduced in the literature, which is kind of shown here. And in our terminology, when we talk about an extracellular vesicle, we are talking about a double lipid layer membrane vesicle, as opposed to extracellular nanoparticles, which we define as being non-membranous in nature. Although, of course, some types of extracellular nanoparticles, like lipoproteins, may have a lipid shell. And different markers have been identified associated with these different populations of extracellular vesicles and nanoparticles. Now, in 2018, the Leiden group published the first paper on exomeres, and they isolated exomeres using asymmetric field flow fractionation. And what they found was that exomeres are non-membranous nanoparticles, approximately 35 nanometers in diameter. And these exomeres were enriched in metabolic enzymes, but they did not ascribe to the exomeres a biogenesis pathway, nor did they ascribe any biological functions. The Leiden group then went on to show that extracellular vesicles and particles combined could be used to define biomarkers for multiple human cancers. And this is an example, I think, of the utility of maybe in some cases lumping together extracellular vesicles and particles for simplicity, and that might yield results in terms of identifying biomarkers. But our approach has been quite different. We have attempted to kind of split apart what's present in the extracellular space. And so our first foray into this field was a publication in 2019 where we provided the first evidence that exomeres could transfer functional cargo. And what we did in this paper is utilize a simplified method of isolation of exomeres based on ultracentrifugation. And what we found was that these exomeres were enriched for organoid proteins and APP. And we also found two different functions, one based on the presence of STGAL1 in exomeres and the other on the presence of AREG in exomeres. Now, then we reasoned that if we could isolate these exomere nanoparticles using ultracentrifugation, would it then be possible to identify or discover other novel types of particles using ultracentrifugation? And here to the left, we see an outline of the approach we used. So at the top, we see that we spin down a crude pellet of extracellular vesicles and particles by using a 4-hour centrifugation spin. And to the left, we then take this crude pellet and further fractionate it by putting it at the bottom of a tube and layering on top a udixanol step gradient. After overnight centrifugation, it's then possible to fractionate the initial crude pellet into its constituent small extracellular vesicle and non-vesicular components, which are enriched in the non-vesicular higher density fractions. But that leaves the supernatant from the initial small extracellular vesicle preparation step. And that supernatant we use to generate the exomeres. These are the nanoparticles that are pelleted from that supernatant with an extended overnight spin. And that was the basis of the paper I mentioned previously. But we then reasoned, well, what about the supernatant that's left over from this exomere purification step? Might that contain types of particles? And what we then did was take that supernatant and expose it to an extended overnight centrifugation step, but this is a significantly higher speed step, so 367,000 G-force. And what we found was that, yes, we do in fact pellet particles, and we dub these particles supermeres from the supernatant of exomeres. We then further characterized these particles and found they are replete with disease biomarkers and therapeutic targets, and they in fact have functional aspects. And so one question that has come up is that, among many questions, is that these supermeres and exomeres, do they represent distinct nanoparticles, or are they basically the same thing? And so here in bullet point, we have what we think are some arguments in favor of their distinction. So we find that exomeres and supermeres have distinct size and morphology, and this has been confirmed now using a ultracentrifugation-based method of isolation. We also found that exomeres and supermeres display distinct uptake profiles in vivo, which has been confirmed by our collaborator, Al Shirest, and I'll go over this uptake in a second. We also found that exomeres and supermeres display distinct proteomic profiles. And additionally, they are also distinct in the terms of the extracellular RNA release and profiles. And we found that when we inject mice with exomeres and supermeres, they elicit differential gene expression in mouse livers. And here we see an example of supermere characterization using liquid phase atomic force microscopy. And the data I'm showing you here is using Diffie cells. That's a human CRC cell line. And most of the data we'll be showing you throughout this presentation is from this Diffie cell line, but we have confirmed the presence of supermeres in the cell culture media from other cancer cell lines and also normal human cells. And we have also been able to detect supermeres in human plasma. So here we see at the top small extracellular vesicles and the non-vesicular fractions as imaged by AFM. And at the bottom, we see exomeres and supermeres. And we quantified and looked further into the morphology, and we found that supermeres are indeed non-membranous extracellular nanoparticles. They are smaller than exomeres, both in terms of diameter and height. So exomeres are typically 30 to 40 nanometers, and 10 to 12 nanometers in height, where supermeres are 25 to 30 nanometers in diameter and 6 to 8 nanometers in height. As I mentioned, we then went on to look at the uptake of extracellular vesicles and nanoparticles in the mouse. So after injection of small extracellular vesicle, exomeres and supermeres, we looked at the uptake in different organs. And what we consistently found was that supermere displayed a greater degree of uptake in all organs examined. But of particular note, what we saw was that in the brain, there was little uptake of small extracellular vesicles and exomeres, but a profoundly greater uptake of supermeres in the mouse brain. And this is, I think, a very interesting result that I'm happy has now been replicated by our collaborator, Algerest. And now I will turn over the presentation to my colleague who will go into supermeres in more detail.
So we further characterize the supermeres by proteomic analysis by LC-MF. So the PCA showed clearly that small EVs is distinct from the other fractions, and non-vesicular and exomeres are more similar. But the supermeres is clearly distinct from exomeres. Although there are considerable amount of overlap between the cargos in exomeres and the supermeres, examination of the differentially expressed proteins by at least 1.5-fold clearly showed that the profile of supermeres is distinct from exomeres. The proteomic analysis data also showed that supermeres are enriched in clinical relevant shared membrane proteins and glycolytic machinery. For example, proteins involved in the Alzheimer's disease, like APP, are highly enriched in supermeres. Other membrane proteins listed here also enriched in supermeres. So metabolic enzymes such as ENO1, ENO2 also highly enriched in supermeres. All these clearly support that supermeres distinct from exomeres. Next, we examine the top 20 most abundant proteins. So FASN, the fatty acid synthesis, is the most abundant proteins in exomeres, while interestingly notice that TGF-beta-I is a TGF-beta induced protein, is the most abundant protein in supermeres. So it has been reported that TGF-beta-I has multiple functions, including metastasis, and also it is a diagnostic and a therapeutic target in multiple cancers. So to investigate the potential clinical relevance of TGF-beta-I in CRC, we have performed TGF-beta-I staining on the CRC TMA. So the level of TGF-beta-I in the CRCs is much higher than in the normal tissues. The level of TGF-beta-I in the is closely correlated with the overall survival of the patient. And also the level of TGF-beta-I in other fractions derived from CRC patient plasma is much higher than in the normal controls, although there's a small size from several patients here. But all these data support that TGF-beta-I is a potential CRC biomarker. So next, we further characterize the supermeres by iron analysis. So the relative iron abundance of the total extracellular RNA in the supermeres is significantly higher than in the exomeres or small EVs. Among all the small RNA populations, microRNA is the predominant RNA species in all the fractions, including the cells. We further examine the RNA sequences, the mRNA and tRNA sequences, it's revealed that the total profile of microRNA and tRNA in the supermeres is distinct from Xmeres, supporting that supermeres is distinct from Xmeres. So next, we examine the top 25 most abundant microRNAs in all the fractions. It's interesting to notice that miR-1246 is the most abundant and also most enriched microRNA in supermeres. So it has been reported that miR-1246 is enriched in exomes and has been reported to be upregulated in several cancers, including lung and liver. To investigate the clinical relevance of miR-1246 in CRC, we have performed FISH. Surprisingly, that the staining intensity of miR-1246 in the CRCs is dramatically high. However, the staining intensity in the normal controls is either undetected or very weak, suggesting that miR-1246 is a potential CRC biomarker. Another surprising finding is that we found that the majority of miR-1246 is in the nucleus, which this promoted us to further examine the resources of miR-1246, the sources of miR-1246. So by examination, the RNA sequences, we find that the majority of miR-1246 sequence are derived from RNU2 families. It's not derived from proposed precursor miR-1246. This is suggesting that the miR-1246 sequence found detected in all these fractions might generated from dicer and torture independent pathway. Dr. Tosar has made many insightful comments about the sources of miR-1246 in his commentary. The high abundance of RNA in supermeres promoted us to further look at the RNA binding proteins. Our proteomic analysis data showed that there are a lot of RNA binding proteins enriched in supermeres. So the Vesicles plot confirmed that some of these RNA binding proteins are highly enriched in both Xmeres and supermeres, including the agonist proteins, XPO5, and also the HNRNP-A2B1. So some of these RNA binding proteins has been reported to be involved in exomal microRNA secretion. So whether they are actively involved in non-vesicular Xmer supermer microRNA secretion need further investigation. Finally, we want to examine the effect of systematic delivery of Xmeres and supermeres on mouse liver. So we found that both supermeres and Xmeres decrease the level of triglyceride and glycogen, and also both decreased phosphorylated AKT and ERK. However, supermeres can selectively decrease liver to body weight ratio, and also supermeres and Xmeres induced marked differences in gene expression by RNA sequencing, which was shown in the right on the heat map. Based on our data, we conclude that supermeres appear to be distinct and functional extracellular nanoparticles, and supermeres are enriched with clinical relevant biomarkers, including TGF-beta-I and miR-1246. So many of the cargo previously reported to be in the exomes or small EVs are highly enriched in the non-vesicular Xmeres and supermeres. So these studies are an important step towards assigning cargo to their correct carriers. Although we have done extensively study on the supermeres and Exomeres, there are still a lot of questions remaining unknown. For example, what is the biogenesis of supermeres and Exomeres? How selective is cargo sorting to supermeres with Exomeres? And the last one is that, is there any further heterogeneity of nanoparticles like the small EVs and exomes? Thank you very much.
Thank you. Thank you to all of the speakers. That was really interesting.
I first want to say that it's really an honor for me to be presenting after this great talk that Dr. Coffey, Zhang, and Jeppesen have provided, and to discuss their excellent paper on NCB last year, full of very important and functional data. And in this commentary that we published in the Journal of Extracellular Biology some months ago, we're only tackling one specific aspect, which is one of the last questions. It's related to the last question that was presented, some of the unknowns, whether exomeres and supermeres are an homogeneous nanoparticle, extracellular nanoparticles, or whether they could be a collection of different nanoparticles. That's what we try to at least place the question here. And I was very interested when the paper appeared, because I have been interested for a while on non-vesicular RNA, extracellular non-vesicular RNA, and it was really amazing to read and learn that most of the extracellular RNA is not associated with vesicles, and we kind of also observed that in our systems, but it's associated with a specific non-vesicular nanoparticles, in this case, the supermeres. And in 2020, we published this paper where we also look for non-vesicular RNA, for carriers, potential carriers of non-vesicular RNAs. And we found that cells release RNA in vesicles, of course, and the RNA which is protected inside vesicles is protected from degradation. But cells also release full-length tRNAs and ribosomes to the extracellular space and outside vesicles. But we could only observe these full-length tRNAs and ribosomes if we inhibited extracellular ribonucleases, because otherwise extracellular RNases will just cleave this the ribosomal RNA in the context of ribosomes. So we are losing these extracellular nanoparticles. So at least here is another example of a different extracellular non-vesicular nanoparticle that is only observable when extracellular ribonucleases are not active. Regarding MIR-1246, it was already introduced nicely in the presentation before. It's a microRNA that we have also been interested at, because it's usually present in a lot of studies where people look for microRNAs that are enriched in vesicles. But it's important to acknowledge that it's a microRNA that is highly abundant in fetal bovine serum, and it's not present in vesicles. It remains in the centrifugation supernatants in FBS, and that's the reason why it tends to appear in many studies where EV-depleted FBS is used. But this does not apply to the paper of Dr. Coffey and collaborators, because they mostly prepare their exomeres and supermeres under serum-free conditions. And then we have this aspect of MIR-1246, that it's possibly or also a fragment of a small nuclear RNA, or RNA-U2. And as was presented, MIR-1246 is interesting because it's the most enriched microRNA in supermeres. And as the authors already acknowledged in their article, it's not clearly whether it's really a bona fide microRNA or a fragment of the RNA-U2. But anyway, it's an RNA which is highly abundant in the extracellular space, and it's highly enriched in supermeres. So that makes it something like we really wonder what was going on with this U2, the small nuclear RNA that is so abundant in these extracellular samples. And let's just remember that whenever our cells transcribe an mRNA, the mRNA matures in the nucleus by a process that we all know is called splicing. And in splicing, we have an adenosine in the intron that attacks the guanosine, typically in the 5' splice sites, forming this intermediate. So this is called the adenosine in the branch site. And this branch site in the introns of mRNAs are recognized, and the region in the intronic mRNAs are recognized by U2 ribonucleoproteins and other small nuclear ribonucleoproteins. But U2 is the one that we care of here. And the U2 ribonucleoprotein is composed of the U2 small nuclear RNA and several proteins. And the core of this complex is composed of 7 SM proteins, which form a ring, which is an heptamer, which is called the SM heptamer, that recognizes a uridine-rich sequence, which is here represented as the SM site. And this uridine-rich sequence is quite characteristic of several small nuclear RNAs. So what we did was we then loaded a cryo-EM structure of the 17S U2 small nuclear ribonucleoprotein to try to identify where this miR1246 region is. And here you can see the heptamers of this SM protein, so the SM heptamer, and in black is the entire U2 small nuclear RNA. And what's represented in red is a region that corresponds or that matches miR1246. And it's pretty evident that miR1246 is actually the footprint of the SM heptamer on the U2 small nuclear RNA. So it's a fragment of the U2 small nuclear RNA that is protected by this complex. Here you can see this ring from a different side. And then we analyzed the small RNA-seq data obtained by Zhang et al. And this is small RNA sequencing that they did. So it's not possible to see reads of spanning the entire length of the U2 small nuclear RNA. But we can see inside cells that there are fragments which span the entire U2 small nuclear RNA in different regions. And the profile in small EVs is very similar to the profile in cells. But then when we go to exomeres, and especially supermeres, most of the reads are only coming from a region which corresponds to this miR1246, or we will call it the SM core region. So it's the region that's protected by this SM heptamer. But the length of the reads is larger than the region which is protected. And our hypothesis is that the entire U2 small nuclear RNA is released into the extracellular space, but then it is cleaved by extracellular ribonucleases in a process similar to what we have seen for ribosomes. And because extracellular ribonucleases tend to be RNase A family member, they cleave 3' to pyrimidines, so U or C. And so if we look at the SM core region, the region which is protected by these SM heptamers, and we look to the first pyrimidine 5' in the U2 small nuclear RNA is here, and the first pyrimidine downstream of this protected region is here, and this matches pretty well with the profile of the reads which are derived from U2 in supermeres. And then if we look in exomeres, we can see that there are more reads which are coming from different regions. So somehow the RNA inside exomeres seems to be somehow a little bit more protected from degradation than in the case of supermeres. But if we have protection by protein binding, then the question was, can we find these U2 associated proteins in the proteomic data collected by the authors? And in fact, the proteomic data supports the presence of all these SM proteins in supermeres and exomeres. So these are the proteins that correspond to the SM heptamer. So 6 out of 7 of the proteins can be detected in supermeres. The only one which cannot be detected is the smallest one, and therefore harder to identify by mass spec. But also other proteins of the U2 ribonucleoprotein, like splicing factor 3A and 3B proteins, can also be identified in supermeres and in exomeres. And although it is incorrect to infer abundance based on spectrum counts, at least we can make the case that some proteins like splicing factor 3B1 seems to be more enriched in exomeres than in supermeres. And this protein is one of the largest proteins of the U2 ribonucleoprotein, which seems to be somehow absent in supermeres. not absent, but less rigid. So our hypothesis is that given the method that the authors are using to collect exomeres and supermeres, maybe the 17S U2 small ribonucleoprotein is pelleting by the G-force that it's used to collect exomeres, and then living in the supernatant, smaller portions of this protein, of this ribonucleoprotein, like the SM heptamers with the sequence which is protected, which will be called MIR1246, and that can be pellet in the supermeres. Then, after seeing this, what about the ribosomes? So we mentioned that ribosomes cannot be detected if extracellular ribonucleases are present, and that's probably the reason why in the Leiden's group's paper on exomeres, ribosomal RNAs were not found. But since the Coffey lab study was performed under certain-free conditions, we wonder whether we could detect ribosomal RNA. And in fact, there is ribosomal RNA in the exomeres, especially. Again, this is small RNA sequencing, so we should definitely not expect reads spanning the reads with a length of the entire 28S ribosomal RNA, which is more than 5,000 nucleotides long. But we can see fragments which are derived from this ribosomal RNA. And when we look at the coverage in cells, we can see that certain regions produce more fragments, especially the 5' end and the 3' end. And the profile in exomeres is very similar to the profile in cells, suggesting that maybe we have also full-length ribosomal RNA in exomeres. In contrast, the profile in supermeres is completely different. So there are no reads in the 5' and the 3' region, and instead, most of the reads are coming from two very specific internal regions of the 28S ribosomal RNA. So very different profiles between exomeres and supermeres, and exomeres quite similar to cells. And then we asked whether ribosomal proteins were also found. And again, looking at this mass spec data collected by the authors, most of the ribosomal proteins from the large and the small ribosomal subunits were present were supported by mass spec counts. But an interesting trend is that the abundance of the ribosomal proteins was always higher in exomeres than in supermeres, except for RPL10A. So there was only one protein which seemed to be more abundant. Again, this is a very sloppy use of the term abundance, but only RPL10A seemed to be more abundant in supermeres than in exomeres. And what's interesting is that if we look at this specific region that produces a lot of reads in the supermeres, and we map that region in the ribosome, in the structure of the ribosome, that region corresponds to the RNA that interacts with RPL10A. So again, a possible an hypothesis here is that given the speeds used to collect exomeres under cell-free condition, ribosomes will be pelleting down and then living in the supernatant some smaller pieces of ribonucleoproteins composed of some ribosomal proteins and ribosomal protein protected ribosomal RNA that could be collected at higher G forces, in this case in supermeres. And then another protein which we found was interesting is galectin-3 binding protein, because it seems to be a marker of exomeres. It's more enriched in exomeres than in supermeres or small EVs. And this protein was also found as enriched in exomeres in the Leiden's group paper on exomeres. So it seems to be like a protein which is involved somehow in exomeres. This protein is also called MAC2 binding protein, and it's a protein that self-assembles into ring-like structures that show a size of 30 to 40 nanometers by electron microscopy. So this protein itself seems to be capable of forming a complex with the size of roughly an exomere. So in summary, one possible hypothesis that could be something that could be going on here is that by differential centrifugation, this is like the supernatant of a small EV preparation. Then by high-speed centrifugation of that supernatant, the exomere pellet seems to be enriched or could be enriched with U2 SNRPs, ribosomes, and decamers of these galectin-3 binding proteins, while the supermer pellet could be enriched with other ribonuclear proteins. So to round up, I can envision three different possibilities, and I really don't know the answer. I just want to pose the question on what are exomeres and supermeres exactly? Are they, so given a supermer, is it a single nanoparticle of a defined molecular composition composed of all these proteins that have been identified and the RNA? Or maybe there is supermer, but then supermer preps are contaminated by other ribonuclear proteins that are also present in the extracellular space, like a ring of SM proteins protecting a region of the U2 RNA, which will be miR-1246 and others. Or maybe supermers are an heterogeneous collection of ribonuclear proteins, and that's not such thing as a core supermer, but it's like what we call large EVs. So large EVs are a mixture of EVs with different composition that we call large EVs because they are collected at a certain G-force. And the same will apply to exomeres. So I think that irrespectively of the case, I think this is a question that is worth pursuing. But irrespectively of the case, I think what I like here is that seems to be like the paper that the Coffey lab published in 2019, showing the different composition that many of the proteins that were assigned to EVs are actually present outside EVs. And now these papers on exomeres and supermeres, they might be like a coming of age for non-vesicular particles and non-vesicular xRNAs, because irrespective of whether they are a single or many nanoparticles, they're clearly functional. And I think that was shown in a very beautiful way. So I expect a lot of new information and surprises once us as a community start searching not only for vesicular RNA, but also for non-vesicular extracellular RNA. So, okay, thanks a lot. And I'm looking forward to continue this discussion with your questions.
This was perfect. Thank you to all the speakers. I think that this was a great start for a discussion. And maybe Ken would want to add something, or was everything said?
No, I think we have a lot of good questions in the chat box. So why don't we go ahead and get started with those? Okay,
perfect. Then let me invite Clotilde Théry. She's an expert in another characterization, not I mean, maybe also for the extracellular, for the nanoparticles, but specifically for extracellular vesicles. She's done a lot of work on that, and I think she'll be great to moderate this discussion. So please, Clotilde, go ahead. And I will just try to ask some questions.
Okay, thank you, Metka. Actually, I mean, you could very well moderate yourself. There are lots of questions in the chat. First, there are questions more to the first presentation. So I think Sayam Ghosal was the first.
Yeah, hi. Thank you for giving me the opportunity to ask the question. Actually, I was a bit curious that we have been seeing that this AF4 technology is one of the methods of characterization of exomere and supermer. So while watching the presentation, I was just a bit curious about this MIR microRNA 1246 and other microRNA profiling. By that, can we distinguish between the exomere and supermer characterization?
So if I understand the question correctly, you want to know if we can use microRNA to distinguish between exomeres and supermeres.
Yeah, exactly.
So while there is some overlap, there's also clearly distinct expression of microRNA in the exomeres versus the supermeres. But especially for microRNA, I think we need to remember that that's very cell line dependent. So we know that different cell lines put out enriched particles for different types of microRNA. So in this diffy cell line, we could clearly distinguish supermeres from exomeres based on the microRNA they carry. But I think you would probably have to do a more thorough study using different cell lines of different origins before you could say anything with certainty whether microRNA is the best way to distinguish between these different types of nanoparticles.
Thank you, Dennis.
Thank you.
Maybe Juan Pablo could add something, if not microRNAs, maybe some of the other RNA structures that you described.
Again, since we only analyze data from Zhang et al. Here, so the same applies regarding the cells of origin. So we are just looking at their data. But yeah, for the ribosomal RNA, for the 28S ribosomal RNA, for instance, we can observe completely different profiles comparing exomeres and supermeres. So in exomeres is kind of the signature that can also be found in cells. So kind of the same fragments which are produced from the large 28S ribosomal RNA are found in exomeres, while in supermeres, many of those fragments are not there, but there are some very specific ones. So yeah, the profiles are different, and they are different at the ribosomal RNA level, and as Dennis just explained, they are different at the microRNA level. So I guess the answer is yes.
Thank you. So the next question is from Anand Sasidaran about TGFBI.
Yeah, hi, thank you. So my first question is towards Dennis. I was really curious to know about the biogenesis, but the speaker said like you guys are still elucidating the biogenesis of supermeres. But my question is, when you said like you could actually functionally induce the phenotype or rescue the phenotype by using supermeres, right? What would be the quantity you'd be required to do this? How much of these supermeres are you going to put inside, and how do you do this? And do you do NTA to analyze them, or do you do something else?
We don't use NTA because NTA isn't really sensitive to particles that are that small. And so the right amount of anything to put in, for example, a mouse, whether it's EVs or nanoparticles, is of course an unknown. So I think for these experiments, we used different kinds of quantities and of exomeres and supermeres, and there's definitely a dose-dependent response in the mouse to treatments. But it's hard. I don't think I would feel confident at this point saying what is the lower and upper limit of treatment with these nanoparticles, because basically we don't really know.
Could you please mention a little bit about more quantification of supermeres?
So for this study, the way we quantified the dosage of exomeres and supermeres was by measuring the protein content, because we know that exomeres and supermeres mainly consist of protein and RNA. So protein quantification was the method we used here, again because it's not really feasible to get a number quantification using nanosite or particle tracking. I mean, the only way we could really look at individual nanoparticles was using atomic force microscopy, but that's not really useful in a routine quantification assay.
Cool. My second question is, like, you guys are overstressed on TGF-beta being a biomarker in supermeres. Do you think is that because of the cell type you have used for your study? If you use a different study, a different paradigm, the component inside the supermeres might be different. Do you think that's the thing?
I think it's highly likely that there's going to be some cell-line dependence, but specifically for TGF-beta I, which is the protein we looked at here, we found it to be the most abundant supermere protein, not only in these diffy colorectal cancer cells, but also in two other cancer cell lines. I forgot which one it was, but it's in the supplementary materials. And a third cell line had TGF-beta I as the second most abundant protein. So it seems like, at least in all the cell lines we looked at so far, TGF-beta I is either the most abundant protein in supermeres or at least at the very top of the list.
All cancer cell lines.
Yes, but we also looked at a normal cell line, and they also, at least that normal cell line, which was a kidney cell line of primary kidney cells, that also had TGF-beta I in it, in the supermeres.
Great, thank you. My last question is to Juan Pablo. John, you spoke about ribonucleotide proteins inside supermeres. Do you think, is it because the abundance of the SM protein, these have been actually packed and been thrown out? Or is there any other mechanism why they're actually specifically targeted towards these small vesicles?
I think this is connected to the unknown on the biogenesis of these particles. So we think that entire components of the spliceosome are present in the extracellular space, either as part of supermeres or either as standalone different nanoparticles. How are they released into the extracellular space? I don't know. There could be some active mechanisms, or it could be that a small percentage of cells that are dying always in culture are releasing just the nanoparticles that they are inside. What's interesting regarding the SM proteins and small nuclear ribonuclear proteins is that people with autoimmune diseases develop antibodies against these SM proteins. That has been known for decades, and it's very well established. And in fact, the components of the spliceosome were discovered by Schwann States and others by looking at the antibodies that people with lupus had against these proteins and the RNA. So this suggests that at least in vivo, under certain circumstances, cells are releasing these components to the extracellular space, either as part of supermeres or either as standalone nanoparticles that co-isolate with supermeres.
Thank you. Thank you all. Beautiful talk.
Thank you. There are lots of questions, actually. Maybe I'll try to put together several. I see two questions on isolation methods and on purity. So one from Federico Cocozza. How did you set the best speed of centrifugation? And one of Qin Zhang. There is no wash step in the supermere isolation step. I'm just trying to put them together. So can you answer these questions about protocol and about washing step and impurity, co-isolation of impurity?
In terms of why we choose to do the steps we did is that basically, since we used G-force of about 167,000 to pellet the exomeres, we reasoned that we would probably use a higher G-force to isolate smaller particles because they would be expected to pellet at a slower rate. So we basically used the rotor we had available that could go the fastest, and that happened to be a rotor that could go about 350,000 G-force. So that's what we initially did, and that's the maximum speed we could use. So that's why we did that. Is that the best way of isolating supermeres? Well, we don't know. It could be that it would be better using higher speeds, lower speeds. We don't really know. And in terms of how do we address the question of is there contamination in our supermere preps? Well, there's two aspects to this. One is, well, what do we mean by contamination? Because we don't really know, as Tosar also mentioned, that what we are calling supermeres could in fact be a combination of different distinct particles. So we wouldn't necessarily know what to look for in terms of contamination. If we think there's true supermeres and something that's contaminating these supermeres, we wouldn't necessarily know how to tell them apart. But I will address the soluble protein question. So what we did, and that wasn't shown in the schematic, but it's in the paper. What we did is that we put that supernatant through a concentration filter, which does not allow the supermeres and exomeres to pass through the filter, but does allow soluble proteins to pass through. So in that way, we try to minimize the amount of soluble proteins that's left in the supernatant, from which we will then go on to purify the nanoparticles. So that's kind of the extra step we do to get rid of most of the soluble proteins.
Thank you. I hope Qin Zhang and Federico are happy with the answer. I'm sorry, I'm trying to rush a bit, but Susmita had another slightly different question. Susmita, do you want to ask your question?
Sure, very thought-provoking research from Coffey Lab. And I was wondering, if you take 100 ml of your culture media or few ml of your serum or plasma, how would be the quantity of protein and RNA distribution between small EVs, exomeres, and supermeres? And that's my question one. And the second question, of course, we have been talking about these byproduct of, the isolation method, free-swimming proteins. And I was wondering if from serum plasma, is there a possibility that the smaller particles shed from platelet-like vesicles in circulation instead of, active biogenesis? Because if there is active biogenesis, you are saying that all these microRNAs and materials, they're packaged specifically in supermeres or exomeres. But I was curious if they are produced, outside of the cells or somehow from the vesicles, from the extracellular materials, so on and so forth. Thank you.
Okay, so to the last question about the platelet-derived particles, I guess the easy answer is we don't know. It's possible that that would contribute some type of nanoparticle to preparations like these. We did not look into that question, but that's definitely interesting. And I think the other question was the quantities of RNA and protein in the different fractions. So what we found is that, as was presented today, was that it seems like the most enriched fraction for total RNA was the supermeres. So supermeres clearly had most of the extracellular RNA, that's total RNA, followed by the small extracellular vesicle, and then exomeres had the least. But clearly, most of the extracellular RNA seemed to be contained in what we are calling supermeres. And it also seemed like supermeres had more protein than either exomeres and small extracellular vesicles. So they seem to be small balls of mostly proteins and RNA as far as we can tell now. And there's the lipid content of both exomeres, but in particular supermeres, is very low. So they do seem to be entities consisting mostly of RNA and proteins.
Thank you. And what about mRNAs? Did you see mRNAs in these RNA materials?
So we did not look at longer RNA. I'm sure that if you look at fragmented messenger RNA, you would probably find some there, but we did not specifically look at that.
Thank you. So there is a question about purity, but and markers for purity, but I'm afraid that what we heard from your previous answers is probably there are no. I just wanted to give the floor to James Patton, who had a question, maybe more specifically on Juan.
Thank you very much. So Juan Pablo, this is a question. So if I'll make a comment first. There's a lot of microRNAs that are in these databases that are not bona fide microRNAs, and MIR-1246 fits that bill. And the protected fragment that you nicely show is from the SM core. So my question is, since 4 other of the snRNAs in splicing also have the SM core, do you find those RNAs in whether supermeres or some other fraction? Do you find them extracellular, protected by the SM core?
Yeah, this is a very good question. It's true that the SM core is not specific for U2. It also works with several other nerves. In the case of U6, they are different. So they are the LSM, but then U1 and U4, they share exactly the same core. So yeah, we went on to look for U4 protected fragments, and we could not find them in the data, at least not at the levels that corresponded to the U2 derived fragments, but there might be a reason for that. And it's the length of the sequence before you find the next pyrimidine out of the SM core that is very long for U2. So in the case of U4, like I don't remember exactly, but maybe 3 or 4 nucleotides out of the SM core, then you have a uridine or acetidine. I don't remember. So we predict that maybe the U4 could be there, but then the fragments which are produced are just too short in order to be sequenced. So the answer is there's some specificity for U2 in the data, but I wouldn't rule out the presence of the others. That might be just, this might be the reason.
Thank you. So I will put together two questions that are related to lipid, the membranes on supermeres, how it was determined that they had no membrane, but this you answered before because they have very little lipids, if I'm not mistaken. But then the question is how, from Navneet Dogra, is why would they be enriched in transmembrane proteins if they have no membranes? So that's for Dennis, and then we will have a question from Metka. I will give you the floor, Metka.
What we see in supermeres is exclusively the fragments of membrane proteins. So we always only see the extracellular part of a transmembrane protein in exomeres. In supermeres. We never see the full-length version of the protein, so not the transmembrane part of the protein. So it's shed membrane protein. So it's not the full-length protein. It's the ectodomain of membrane proteins.
Okay, thank you. I think this answers. Metka, did you want to ask your question?
Yes, I actually have two questions, but the other one I had to hide behind the first question. So the first one is about the supermeres. If I understood correctly, they can pass the blood-brain barrier while exomeres cannot. And I was just wondering, is this just due to the small size of supermeres, or is there any specific molecular composition that helps with that? Or
So our initial thought was that it's probably because of the size difference, but the size difference is not that large. So can it really account for this very big difference in the uptake to the brain? And this is something that's under active investigation together with collaborators. And we think, no, it's probably not just due to the size, but we don't have any further details of that, but except to say it's probably not due only to the size difference.
And my other question is actually related to lipoproteins. So you hear a lot that, reported a lot that lipoproteins also carry microRNAs or small RNAs. And I was also always wondering, is this maybe just because some other impurities or small particles like RNA particles or exomeres or supermeres were actually co-isolated with these small lipoproteins, so that, you know, is it really shown that, was it really shown that microRNAs directly bind to lipoproteins, or is it just that before people didn't know that these other nanoparticles exist and didn't look for them? So, yeah.
So I didn't do that research myself, but I know that people here at Vanderbilt have looked at that, and I believe that they specifically did pull down assays of high-density lipoprotein and found microRNA associated with that. So they didn't do like a crude bulk purification of nanoparticles, including HDL. They specifically pulled down HDL, and when they do that, they can still find microRNA attached to HDL. So probably HDL does, in fact, carry some microRNA. And I believe the same is true for the larger lipoproteins, but we did not specifically look into that.
Okay, thank you. Thank you for clarifying that. And I think that we'll just have time for two short questions. And there is one question about from Navneet Dogra about a mechanism of internalization of these supermeres or exomeres. Is anything known about that?
Yeah, so we had that in the paper where we looked at uptake and in vitro, and we did some different drug treatments, and it seems like macropinocytosis is probably a main pathway of internalization of exomeres and supermeres. So we can't say at this point precisely whether there's more specific types of uptake going on, but it might be a matter of bulk uptake. So you have engulfment of the extracellular environment, and that internalizes the exomeres and supermeres, because in contrast to the exomeres and supermeres, we saw that small extra vesicles were taken up considerably faster than both exomeres and supermeres, probably indicating that you have some kind of receptor-mediated internalization of the small extracellular vesicles based on their surface protein, but that might not be the case for exomeres and supermeres.
Okay, thank you. And I saw that actually the last question is by Susmita to Jeff, who answered about the platelet poor plasma. So Jeff, if you're still here and you've seen the question.
Yeah.
Okay, perfect. So it's about, are you saying that PPP did not pass on the platelet markers to subsequent smaller sized vesicles? And is it possible to run flow cytometry analysis of supermere fractions with confidence? So if we have you here, please.
Yeah. So, I mean, I would leave the flow CD41. CD41B. CD41. So I'll leave the flow questions up for Jim to answer, but in terms of supermeres and exomeres, but we tested the EVs with CD41B and using our procedure, which is using buffered citrate, we did not see platelet contamination. Yeah, so that's how we kind of assayed for that in the past for our collection.
Okay, maybe a quick question on the last, quick answer on the last question from Nigel Kurgan. He was interested in what are the time points of in vivo injection and how did you determine these time points? Do you think this explains difference in BB crossing? So this is about biodistribution and going to
let's see if I can remember. I think we looked at the time point that was 24 hours at the most. So it's a time-dependent mechanism where if you bury the time point at which you're looking at the organs, would you see a distinction, meaning that maybe what we're observing is that one nanoparticle is simply being taken up at a different rate than the others. I don't think we can say that at this point. It's definitely something that would warrant further investigations and do a larger study using more time points, because that's definitely an interesting possibility.
Okay, so I think that we should stop here because we're already 5 minutes past the time, but this was really an exciting exchange of opinions. And I would like to thank Robert, Juan, Qin, Dennis, and also Ken for really doing an excellent work in presenting and discussing your work. And of course, Clotilde, who did a magnificent job for moderating this discussion. And of course, everyone that joined us today, you are very welcome to join us also next week for another interesting EVClub and another discussion. So thank you all for joining and have a great day.